All right, hi guys, this is a review of Physio for the sleep and wake cycle. This is an extremely long lecture that we were given, so we're going to try our best, but we'll just go through trying to understand sleep/wake cycle. Starting off by, just a reminder, we had in the previous lecture talked about the thalamus and cortex loops that are believed to be the basis for psychological aspects of brain function, so perception, thinking, memory, executive functions. These are the outputs of these thalamocortical loops. As our thalamus is our relay stations connect to all parts of the cortex. All right, and so the idea is that TC loops involving specific pathways give the content to consciousness. And these are like the vision, hearing, somatosensation, executive function, motor output. Then the TC loops involving non-specific pathways cause the brain to be conscious to various degrees. So like fully awake, tired, conscious, or unconscious, or coma. So TC loop specific give content to the consciousness. Unspecific thalamocortical loops give us consciousness overall. Okay, so then the specific thalamocortical loops that are doing something with vision, hearing, smell, sensation, executive function, or motor output, you have general pathways, some sort of sensory area, some sort of executive control area, right? Think frontal cortex, and then some sort of motor output area. All right, but there's memory systems for each of the major functional division. The sensory memory is declarative memory that's fact or event-based, that's formed in the hippocampus surrounding the temporal lobe. And then you have executive function memory, which is generated by the prefrontal cortex, and it's called working memory. And then finally, motor or procedural memories are learned complex patterns of muscle movements, and it's stored across the motor system, but think, talking, driving, moving, etc. But we do have our, three overall like states of like global brain states. We have being awake, waking, we have non-REM sleep, and then we have REM sleep. And so the thought is that we're going to have this reticular activating system is going to cause the thalamocortical loops to follow this these three states, transition between waking, non-REM, and REM overall. All right, but this is just a quick diagram saying between the thalamus and the cortex you have these thalamocortical loops, and then you have back and forth. You have sensory memory being formed in the hippocampus, executive memory being executive decision making, planning. That's the prefrontal cortex being stored there, and then motor or procedural memory being stored in the motor system. And then we have sensory input is actually optional to the system, and motor output is optional to the thalamocortical loop overall. And then you have this reticular activating system, which determines whether or not the system is awake or asleep. All right, so, but then the sleep cycle itself, it's in alternation between non-REM and REM sleep. It occurs roughly every 90 minutes. Non-REM sleep has 4 stages. It's also called synchronized or slow wave sleep. The 4 stages are, stage 1 is marked by alpha waves. And so you may remember we had gamma, beta, alpha, and then we had delta was our last one, and theta waves was in between that, and alpha waves were sort of our like starting to relax brain waves, and beta waves were like the anxious one or alert. So stage 1 is the ECG is marked by alpha waves. You have decreased awareness. You're starting to go to sleep. Decreased awareness is sensory input, relatively easy arousal. Then stage 2 of NREM is marked by sleep spindles, which are high frequency bursts, and K complexes, which are sudden large amplitude waves. You have a decrease in body temperature, breathing, and heart rate, and more disassociation from sensory stimuli. So as you get further into the stages of NREM, you are less and less sensory information is getting to your TC loops. Okay, then stage 3 is associated with delta waves for the first time, and then you still have the sleep spindles, which again, short high frequency bursts, but delta waves are very low frequency brainwave patterns. We said less than 4 hertz was the frequency of those, and they're evidence of pathology if you're awake and you have delta waves, but if you're asleep, they're considered normal. But blood pressure falls, body temperature falls, breathing slows. Stage 4 ECG has the greatest amount of these delta waves, again, super low frequency waves, and then it's a very high arousal to wake up, but you also have the release of growth hormone. So why do you have these delta waves? Well, you may remember that we had the thalamus has two different modes. You have the burst mode that's associated with sleep, and then you have the tonic mode, which is associated with rest. And so burst mode causes mass synchronization of cortex neurons. And if they're all in sync, they're going to have high amplitude, low frequency waves, and those are delta waves. Okay, so then we had random eye movement sleep, REM sleep, and this is also called paradoxical sleep or desynchronized sleep. And REM EEG shows beta waves, which are like anxious or alert, brain waves, alpha waves, which are relaxed, theta waves, which are like you're asleep, and then the REM EEG looks, because of these beta waves and these alpha waves, looks more similar to waking or NREM stage 1 overall. But in REM in particular, you're going to have peripheral atonia. Atonia means no muscle tone. You're going to be paralyzed, with the exception of eye movement, middle ear muscles, and you're capable of breathing. And of course, your heart is beating. But the motor cortices in the brain are highly active. However, their synapses in the ventral horn of the spinal cord, we'll see, are in fact inhibited. So you are have peripheral atonia. Your extremities are locked in. All right, so then also during REM sleep, you have highly erratic autonomic behavior, loss of thermoregulation, high blood pressure, or blood pressure rises, increased breathing frequency and regularity, and then if you're a male, you could have a penile erection. This is associated with the tonic mode of the thalamus, just like being awake is associated with the thalamus. All right, so then again, this is just showing that, you know, every sort of 90 minutes you're cycling from stage 1 of non-REM, stage 2, stage 3, stage 4, and then you get to REM, but during REM you sort of ramp back up to non-REM, and then so now you're non-REM, and then back down to REM, and it repeats over and over again, but the REM part of this gets shorter each time. All right, so actually this is the part that is REM right there, this duration. Okay, so this one is just talking about the stages of sleep, and I thought it'd be fun to listen to sleep sounds. So let's see, 1 second. All right, this is so dumb. Okay, so stage 1, as we begin to nod off, we fall into the first and lightest phase of non-REM sleep. The senses switch off. We no longer hear the light snoring of our spouse, nor do we feel the chill of air coming through the window, but we can easily be aroused by the neighbor's screeching car alarm. So stage 1 is marked by low frequency alpha waves. All right, low frequency alpha waves. Then we get to stage 2. And in stage 2, our breathing and our heart rates even out. Our temperature drops, and we become increasingly disengaged from our waking senses. Marker is brain waves alternate between short bursts called sleep spindles and sudden large waves called K complexes. Sleep spindles. All right, listen to the relaxing music. This is your subconscious speaking. All right, here we have a K complex. All right, then we get to stages 3 and 4 of NREM. In the deepest phases of sleep, also known as slow wave sleep, blood pressure falls, breathing slows, and body temperature drops even lower. These phases are essential for storing energy, strengthening the immune system, and prompting the body to release vital growth hormones. The marker for stage 3 and 4 are wide, slow delta waves. All right, notice how the delta waves are very high in amplitude, and they are relatively infrequent if you compare them to the alpha waves of stage 1. Then we get to REM. Your eyes dart back and forth, breathing and heart rates flutter, and muscles are mostly paralyzed. While we vividly dream of performing gold medal dives at the Olympics, portions of our brains may focus on real-world pursuits, solving problems from the previous day, and burning knowledge and experience into long-term memories. And our marker for this one is beta waves, which we can see here, high frequency, almost as high frequency as gamma waves. All right, so hopefully we had the relaxing sounds of ocean that help us take us through the sounds of stages of sleep. All right, so then we have, moving on though, we said that the reticular activating system helps determine whether the system is asleep or awake, these thalamocortical loops. And so, again, remember there's two types of pathways in the brain. We have specific pathways, right? Think the hearing, or in this case, retina, thalamus, visual cortex, and we have the non-specific pathways. And so these non-specific pathways are called the reticular activating pathways, RAS. They originate in the brainstem, cerebrum, or diencephalon. We love the diencephalon, thalamus, hypothalamus. And then how many functions, or they have many functions, but importantly, they regulate the sleep-wake cycle. And then importantly, we have a number of different nuclei for the reticular activating system that are important because we're going to define them as either pro-sleep or pro-waking. Right? It turns out there's not that many that are pro-sleep, so most of them are pro-waking, but let's just start off by talking about the first one. So the first one is the locus coeruleus, which we'll abbreviate as LC in the future. It produces neuroepinephrine, and that's located right here. And notice this is the pons. Okay, so we have the locus coeruleus in the pons. Its function is phasic and tonic excitability of the cortex to maintain a waking state. Then we have the production of dopamine via the basal ganglia nucleus, which is the nigrostriatal. Then we have the mesocortical and then the tubrohypophyseal. They all produce dopamine. And nigrostriatal has roles in cognition, motor output, phasic, waking, motivational. Mesocortical is going to regulate frontal system activity. And then tubrohypophyseal inhibits the release of prolactin. So these ones actually we don't need to know too much about for this particular lecture, but it's good to know. And then histamine, we have the tubomammillary nucleus of the hypothalamus. This is going to produce histamine. And so you can see here, posterior hypothalamus, and we have this tubomammillary nucleus. And then we have the raphe nucleus, which produces serotonin, and it functions in waking. And so you can see that here, dorsal raphe, located in the rostral pons. And then acetylcholine is going to be produced by the basal forebrain, which is very important. And then the pontine lateral dorsal tegmentum, which we can see here. And then the pedunculopontine tegmental, aka PPT nucleus, and the functions of these guys are going to be controlling the excitability during both waking, the basal forebrain, and then sleeping, the lateral dorsal tegmentum, and then the pedunculopontine tegmental nucleus. All right, so these two, LDT and PPT, the lateral dorsal tegmentum and the pedunculopontine tegmental nucleus are going to be pro-sleep, and then the basal forebrain is going to be pro-awake. All right, so then we have the first model of going from waking to sleep was the reciprocal innervation model. And we were told this model because it is very similar to the current model. It's like almost correct. We have this nucleus, locus coeruleus, and on the previous page we said it produced neuroepinephrine. And then we have the dorsal raphe nucleus, DR, which produces serotonin. Then we also have the pedunculopontine tegmental nucleus and the pontine lateral dorsal tegmentum, which release acetylcholine. Okay, so with that in our head, basically the idea is that these two nucleus, locus coeruleus and dorsal raphe, during waking are active, and they inhibit the pedunculopontine tegmental nucleus and the lateral dorsal tegmentum nucleus. Okay, so these two guys are going to be pro-waking, and then these guys are going to be pro-sleeping. So during waking, your pro-waking nuclei are active, and they inhibit your pro-sleeping nuclei. All right, and then during sleeping, you have the pro-sleeping nuclei are active, and they inhibit your pro-waking nuclei. All right, so the locus coeruleus and the dorsal raphe are pro-waking nuclei. All right, and so the model is basically saying during waking, your locus coeruleus and your dorsal raphe are active, so you have a lot of serotonin, norepinephrine. So you have dorsal raphe is producing serotonin, locus coeruleus is producing neuroepinephrine, so those are both high because during awake, the pro-awake nuclei are active, and they're inhibiting the pro-sleep nuclei, in this case, PPT and LDT, okay? So during wake, those guys, acetylcholine is going to be low. During non-REM sleep, they'll about be equal, and then during REM sleep, the pro-awake nuclei are inactive, the pro-sleep nuclei are active, and so you have the opposite true, which is your PPT, LDT nuclei are active, and then your locus coeruleus and dorsal raphe nucleus are inactive. Okay, so basically the idea is during waking, your pro-awake nuclei are working, they're active, they're inhibiting your pro-sleep nuclei. During sleep, your pro-sleep nuclei are active, and they're inhibiting your pro-awake nuclei. And so this is pretty close to what's actually happening. It's a simplified the reciprocal innervation model was a little bit too simplified because there's more brain regions involved in the control of sleep and wake. And then we also found out that the PPT and LDT, the pedunculopontine tegmental nucleus and the pontine lateral dorsal tegmentum nucleus, which are pro-sleep, they actually don't inactivate the locus coeruleus and dorsal raphe nucleus, which are pro-awake. They actually activate them, which is really bizarre, but it's going to be giving us the reason that REM sleep looks so much like being awake. Okay, so that brings us to the current model of sleep. And the reason that we had introduced so many of these different nuclei over here, right? We said there's neuroepinephrine, we got dopamine, histamine, serotonin, and acetylcholine, and there's like a whole bunch of different nuclei, and it's like overwhelming and awful. And you know, so why do we need all this? It's because this particular model of just the locus coeruleus and the dorsal raphe and the pedunculopontine tegmental and pontine lateral dorsal tegmentum are not quite sufficient. So we have more nuclei that are involved, and so how do we actually deal with them? We separate them into what I had already started to say, which is we have the pro-awake nucleus and then we have the pro-sleep nucleus. Okay, so our current understanding of brain circuits dealing with the sleep-wake cycle involving brain regions or involving sleep-wake is just you have the pro-awake nucleus and the pro-sleep. So basically what we've done is we've put a plus next to it if it's a pro-awake nucleus, and then a minus next to it if it's a pro-sleep nucleus, and then we have categorize them based on where they are. So we have the brainstem nuclei, the hypothalamus nuclei, then we have the basal forebrain area, which doesn't have a specific nuclei there, but it's just the basal forebrain as an area is involved with sleeping and its pro-arousal. All right, so then we have the brainstem nuclei. So the brainstem nuclei, we have locus coeruleus, which produces neuroepinephrine. We're familiar with it. At least we've been familiar with it for about 2 minutes now. We have the dorsal raphe nucleus, which produces serotonin. So dorsal raphe here, and then let's see, locus coeruleus right there in the pons. All right, then you have your pro-sleep nuclei, which were the ones we're familiar with, the pedunculopontine tegmental, and then the pontine lateral dorsal tegmentum, which produce acetylcholine and mediate REM sleep. And so that's located right here and right here. Both produce acetylcholine, both are pro-sleep. All right, then we're moving up. Now we have the hypothalamic nuclei in the hypothalamus, and we have VLPO, ventrolateral preoptic nucleus. And you're like, how can I ever remember this? And I don't think you necessarily have to remember every single thing here. It's just, we want the pro-sleep and then we want the non-pro-sleep, okay? So you're good. All right, we're all good here. Then we have, but LPO, lateral ventral preoptic nucleus, causes sleep. Then we have TMN, tuberal mammary nucleus, which produces histamine. This is pro-awake. And then we have the lateral hypothalamic nucleus, which produces auroxin. This one, auroxin, is going to stabilize during the awake cycle. The wake part of wake sleep cycle is going to stabilize the other arousal nuclei. All right. Then we have the basal forebrain, which is like located like right here, and it is going to release acetylcholine during waking. It's going to inhibit this pro-sleep nuclei, so VLPO, which causes sleep overall, and it releases GABA and galenin. But the basal forebrain is going to cause the waking state, and then VLPO is going to overall cause sleep. Okay, so basal forebrain, so this one's actually important to know, and then basal forebrain is important to know. Okay, so basal forebrain is going to inhibit this guy, and then finally we have this chemical adenosine, which is our somnogen, just a general term for like substance that puts you to sleep. And so the idea is adenosine is going to be pro-sleep. It builds up over the course of the day in the cerebral spinal fluid, and it's going to inhibit the basal forebrain. Okay, and if the basal forebrain is pro-sleep, then it is going to cause you to get tired and sleepy. All right, so we have adenosine as not a nucleus, but instead it's just a chemical that is going to inhibit the basal forebrain, which is ultimately responsible for causing waking, and then that basal forebrain is going to inhibit VLPL, which does in fact cause sleep. And then all these other guys are going to be pro-sleep in the case of locus coeruleus and dorsal raphe, and then in the case of PPT and LDT, pedunculopontine tegmental and pontine lateral dorsal tegmentum, those are pro-sleep. Okay, so then what's the actual sequence? Because we've said a lot of words, and honestly, it's like blah, right? So we're trying our best here. We have, what are the sequence of events causing sleep? So I have written them all out here, but this chart we have here is actually quite good. So we'll take a look at the chart, but first off, waking, basically, if you're awake, the pro-awake nuclei are going to be active, okay? So what does that mean? We have the locus coeruleus, we have the dorsal raphe. Those are going to both be active, and they're going to go ahead and inhibit, just like in our original theory of sleep, right? Those two are going to, locus coeruleus, dorsal raphe are going to inhibit PPT and LDT, and remember that's pedunculopontine tegmental and pontine lateral dorsal tegmentum. Yikes. All right, so those two are going to inhibit our pro-sleep ones, and then we have TMN, tubero-mammillary nucleus, is going to release histamine. This is pro-awake. And then we have lateral hypothalamic nucleus is going to release auroxin, which is going to stabilize all three of these guys, okay, such that they're not inhibited. All right, and then finally, we also have the following are inactive during awake stage. So VLPO, ventral lateral preoptic nucleus, and the hypothalamus, which causes sleep, is going to be inactivated by the basal forebrain. All right, the basal forebrain itself is active right now because you don't have very much adenosine at the beginning of the day, right? Or when you're around, it builds up throughout the day. All right, so then what does that look like here on our chart? So we have, here's our pro, and pro-awake nuclei, right? That's what we see here. And then another pro-awake nuclei. This one is going to release auroxin, which stabilizes these guys. Locus coeruleus and dorsal raphe are going to inhibit our pro-sleep nuclei. And then the basal forebrain is going to inhibit our pro-sleep nuclei in the hypothalamus, VLPO. During this waking time, though, you also have the slow accumulation of adenosine in our cerebral spinal fluid, and adenosine is eventually going to get to that basal forebrain, and then this guy is no longer going to be inhibited, and then this guy is going to go ahead and inhibit the other stuff. But we're not there yet. So next is the accumulation of adenosine in the basal forebrain. So this is going to shift the brain towards being pro-sleep. Adenosine inhibits the basal forebrain, and so now the ventrolateral preoptic nucleus of the hypothalamus, which causes sleep, is less inhibited. All right, so the basal forebrain is inhibiting this guy, but not if adenosine inhibits the basal forebrain, right? So now the VLPO is going to be able, because it's less inhibited, it's now going to be able to inhibit the pro-awake nuclei, and so now the locus coeruleus, dorsal raphe, tubero-mammillary nucleus, the lateral hypothalamic nucleus are all going to be inhibited, right? And so now we have inhibited all of our pro-awake nucleus as a result of this adenosine accumulating and knocking out, inhibiting our basal forebrain, and now our basal forebrain can't inhibit this guy, and so now that this guy is active, it's going to go ahead and inhibit our pro-awake nuclei, and if our pro-awake nuclei are inhibited, then there's nothing keeping us awake, and VLPO wins, and now we are asleep, okay? So let's just take a quick look at that. So we have adenosine is reached its kind of its peak, and it's going to decrease throughout the night, as we'll see in just a second. But its high value is inhibiting the basal forebrain, which is pro-awake. VLPO is now no longer inhibited, so it's going to itself inhibit the pro-awake nuclei. So this guy's inhibited, you get inhibited, you get inhibited, you get inhibited. Okay, and so if locus coeruleus and dorsal raphe are inhibited, then they are no longer able to inhibit PPT and LDT, which are pro-sleep nuclei, okay? So you can kind of see how adenosine has gone and screwed everything up. So now we are pro-sleep. Actually, it's not screwed up because it's a good thing to be asleep. All right, and we're like, you're putting us to sleep. Yeah. All right, then we have NREM passes through stages 1 through 4, right? So as you go further and further into sleep by increasing inhibition of the pro-awake nuclei. So locus coeruleus, dorsal raphe, tubero-mammillary nucleus, and lateral hypothalamic nucleus. And then eventually LC, locus coeruleus and dorsal raphe get so inhibited, they no longer inhibit the LDT and PPT, which again are pedunculopontine tegmental, PPT, and pontine lateral dorsal tegmentum, which are the pro-sleep. And so these guys have become active, and once you get to the stage where these guys are fully active, you've now transitioned from stage 4 of NREM to random eye movement sleep. Okay, so now we're here. And when we're in REM sleep, remember, the original thought is if you have in REM sleep, the original theory was that if these guys are active, they're going to inhibit these guys. And is that what I just said? I think I might be getting too tired. I'm so sorry. That was the original theory, that these guys being awake was going to go and inhibit the pro-awake ones. This one, VLPO, will inhibit our pro-awake nuclei. I'm so sorry, guys. I've been at this too long. When our PPT and LDT are no longer inhibited, as is in the case of REM, they are not going to go through and inhibit locus coeruleus and dorsal raphe. In fact, they go through and stimulate these guys, okay, which is very bizarre. Because it's not what we would have thought. So during REM, the pro-wake nuclei are maximally inhibited, and then LDT and PPT ironically activate locus coeruleus and dorsal raphe, despite the fact that they are pro-awake. And then this will cause a shift back to NREM sleep, but eventually locus coeruleus and dorsal raphe will become inhibited again, and the brain goes back to REM sleep. All right, so you go from REM to non-REM based on the fact that the PPT and LDT are going to stimulate these guys, and then the VLPO is still going to inhibit all of these guys, right? So you get kind of a tug of war going on, and as a result, you go from REM to non-REM overall, but PPT and LDT are going to cause you to go to non-REM as a result of the locus coeruleus and dorsal raphe being stimulated. All right, but specifically, these two guys, LH and TMN, do not get stimulated, so they're still like asleep until the adenosine goes away and the basal forebrain is back in play. All right, so then eventually during sleep, adenosine does get depleted, and the inhibition on the basal forebrain is lifted. As a result, the pronuclei, in particular VLPO, is inhibited, and then PPT and LDT are going to be inhibited as well. And as a result of all of our pro-sleep guys being inhibited, our pro-wake guys are no longer inhibited, and you go ahead and wake up. All right. And then just a quick point that there's the hypothalamic circadian rhythm circuits, and these work in conjunction with the pro-wake nuclei to keep them active, right? So you wake up at a certain time of the day. All right, so we are almost done. It doesn't look like it, but so then we have this concept of being paralyzed during random eye movement, and we have this EEG, which doesn't quite seem to make all that much sense. So why is that the case? Well, we have during REM sleep, we have peripheral paralysis, atonia. It's brief, but it's intended to keep you from acting out your dreams. And so the mechanism is REM sleep leads to the activation of pedunculopontine tegmental, PPT, and pontine lateral dorsal tegmentum, LDT, which produce acetylcholine. And then the PPT and LDT are going to activate the perilocus coeruleus, which is adjacent to the locus coeruleus, and then that's going to activate a medullary nucleus with glutamate, and then the medullary nucleus is going to project to and release glycine, which is inhibitory, into the ventral horn, which is going to inhibit the motor neurons in the ventral horn. And so as a result of PPT and LDT activating the perilocus coeruleus, so adjacent to the locus coeruleus, that perilocus coeruleus is going to cause a medullary nucleus to project to the ventral horns along the spinal cord and inhibit the motor neurons there. And as a result, any motor commands that are coming from the cortical spinal pathway are going to be shut off. Okay, so this is what it's saying. This diagram right here is we have acetylcholine coming from our pedunculo, basically LDT and PPT, and they're going to go ahead and give acetylcholine to the locus coeruleus, which is going to send an axon down and synapse on this medullary nucleus, and then the medullary nucleus is going to send an axon down to the ventral horn, and then synapse on the motor neurons there, but it's going to do an inhibitory synapse to keep those motor neurons from discharging and causing the muscle to contract when you do get a signal from the cortical spinal pathway, the voluntary muscle movements. All right, so as a result, you'd be paralyzed during REM sleep because you have PPT and LDT are activating this pathway. All right, so then what's going on with the EEG during REM? Because you have, this is your awake EEG dominated by beta, right? And then here is REM, and then here's non-REM. So this guy is the odd one out. So awake and REM look very similar. Why is that the case? Well, it turns out that acetylcholine from the PPT nucleus, the pro-sleep nucleus, inhibits the thalamic reticular nucleus. And you may recall that the thalamic reticular nucleus is kind of like the rhythm nucleus of the thalamus. And so it sets sort of like the overall rhythm at which the relay neurons interact with the neurons of the entire cortex, whatever cortex neurons they're synapsing with. And so if PPT inhibits the reticular nucleus, then the thalamic relay nucleus are no longer inhibited. They're therefore activated, And if they're activated, we are now in tonic mode, okay? So tonic mode is awake mode, and we're in awake mode despite still being asleep, because you may recall PPT during REM is going to act on the locus coeruleus and the dorsal raphe, which are pro-awake, but the other, the basal forebrain is still asleep, right? Still inhibited by the fact we have a bunch of adenosine, and then you have those other two nuclei, LH and TMN, which are lateral hypothalamus, and then TMN was our tubulomammary nucleus, and those two are like still going to be inhibited. So you're still asleep, but your the PPT has activated or inhibited the thalamic reticular nucleus, and it's going to mean that the thalamic relay neurons are no longer inhibited, and therefore you're in tonic mode, which is like the awake mode of the thalamus, despite the fact that the rest of the cortex is not awake. All right, so the acetylcholine also activates the cortex neurons, and so the thalamocortical loop is now active with no sensory input because you're asleep, and then the EEG, therefore during REM, is going to look like the EEG because you have these thalamocortical loops, which when they're running, they produce our waves that when you're like conscious are going to be like beta for alertness and anxiousness, and then alpha when you're like resting. All right, so remember that REM for the thalamus, thalamus is in tonic mode, and the non-REM thalamus is in burst mode, right? But tonic mode we associate with being awake. All right, so then this was a particularly interesting thought. So forms of mental activity during sleep. So we normally associate sleep with unconsciousness. However, it only appears this way because we don't easily remember conscious experiences that occur during sleep. What? But lack of remembering is not the same as lack of consciousness. Okay, so you can be conscious during sleep, and so people with sleep disturbances may readily remember different forms of sleep consciousness. So what are the different forms of sleep consciousness? What are the mental states you can have during REM and non-REM? All right, so non-REM, you have some different conditions. So you have hypnagogia, which is associated with stage 2 non-REM. You have very vivid perceptions, usually visual, but can be auditory, tactile, and these perceptions do not occur in a unified sensorium. For instance, you may see isolated visual images set in a black field. All right, so here, like you get flashes of light, floating or drifting faces, nature scenes, auto-symbolic phenomenon, or just like bizarre. Like there's still some consciousness there. We remember as conscious experience, despite the fact that you are asleep, right? Okay, and so this issue is like you can remember this, whereas maybe you might actually experience this normally. All right, then we have sleep terrors occurs during non-REM sleep, little apparent content to consciousness, characterized predominantly by non-specific feelings of terror, no associated perceptions. Then you have NREM mentation, so lacking rich sensation, no perception of sensory environment. Upon waking, person reports they lay there thinking. The thought is repetitive, banal, or nonsensical. And these occur when the thalamus is in burst mode. So burst mode prevents communication with the senses, but does not prevent cognitive activity. And then sleepwalking cannot occur in REM because of paralysis. So these are non-REM phenomenon. No perceptual, emotional, or cognitive content. Sleepwalkers are unaware of the external stimuli, although their eyes are open and face is expressionless. Expression of complex motor activities in the absence of any mental activity resembles automatisms of epilepsy. And then 1 to 5% of adults with a higher incidence in children, and it's associated with a delta ECG that precedes delta wave in the ECG that precedes movement. All right, so then REM mental states, you have non-lucid dreams. Person is not aware they're dreaming. There's a lack of access to voluntary recall. Submersion in a complete sensorium. All sensory modalities are present. It occurs in 80% of REM awakenings, 25% in non-REM awakenings, characterized by formal bizarreness, discontinuous scene changes, no real perceptions, characterized by high incidence of anxiety producing content, being attacked by animals or other humans. And then lucid dreams, person is aware they're dreaming, implies access to voluntary recall. Otherwise, just like non-lucid dreams, it's common with narcoleptics because they can transition directly from waking to REM sleep. And then sleep paralysis occurs at REM to waking transition. The mind wakes before the body, and the person is aware of REM atonia. It can occur in a dream immediately preceding to awakening. And then REM and tonic mode. So what function is served by the brain going to tonic mode during sleep? No one knows for sure, but believes it had an evolutionary advantage for rehearsing dangerous situations. And then neurotransmitter balances prevent sensory input and motor output from passing through the thalamus, but higher order associative cortices are highly active during REM. All right, cool. So yeah, that was a lot, and we're on our last page, so we're just about done. So just a quick summary of mental function across waking and sleep. Waking, cognitive functions work together in a smooth, coordinated way. The thalamocortical loops are dominated by tonic mode. Hopefully, that's like one of the few things we get from this. Waking, tonic mode, REM, tonic mode, non-REM, burst mode. Got it. Free question on an exam, hopefully. Then you have, during waking, we have binding together to like form our quote-unquote consciousness of all psych functions via gamma waves. Binding is very strong during waking. Non-REM binding is absent. It's dominated by burst mode. Sensory input is absent. All content and consciousness is hallucinations. And what are hallucinations? They're conscious awareness of something not presented to the brain by the senses. And then finally, we have REM, where you do have binding coming back. So you have presence of more gamma waves in your EEGs. Tonic mode dominates in the thalamus. Psychological functions are coordinated, but you still have no sensory input or motor, and you have no motor output. Hallucinations called dreams occur, and you have no executive action, no output, but you can have lucid dreams, which means you have access to long-term declarative factor event-based memory, and you know that you're in a dream. And then you have non-lucid dreams where you don't have access to these declarative memories. All right, so this is just saying, like, here's the diagram. Waking, your tonic mode, binding is strong. We have a lot of these gamma waves. Non-REM, you're in burst mode, binding is not there. Weak or non-existent, only one function is expressed at a time. For consciousness. And then REM binding is more present, but you have no sensory input, and you have like bizarreness. All right, so then sleep disorders, sleep apnea, frequent stop of breathing, obstruction of airway. You can see that here. Most common in overweight males who snore. Bedwetting, fairly common, associated with NREM sleep for children. If you're in stage 4 of NREM, which is where this is associated with it, then often in REM dreams immediately following that, you have the sensation of wet clothes, which is not fun. And then finally, or second last, you have REM sleep behavior disorder, RBD, characterized by the atonia of REM sleep, but you have the appearance of talking or movement. This is characterized with a loss of atonia during REM sleep. So you have REM sleep, but you do not have inhibition of your muscles, so you can still talk and move. And this is thought to be due to diffuse lesions in the brain or brainstem, but it's readily treatable with a benzodiazepine clonazepam. All right, then finally you have insomnia. It's very present, 5 to 10% of adults in Western industrialized countries. Trouble falling asleep, trouble staying asleep, stronger risk for depression. You have 2 types. You have primary insomnia due to psychological hyperarousal processes, so think something wrong with the brain. And then a secondary is triggered and maintained by illness, drugs, alcohol, etc., more lifestyle things. All right, so that was it. Holy cow, that was long. So hopefully that was helpful. It was helpful for me. But I hope you guys are doing well and not stressing too much. But yeah, so good job, guys, and keep at it.